Hello students. So today we will discuss about some numericals on Meselson and Stahl's experiment. Okay. So in the competitive examinations, so some problems will be asked on this experiment. To solve those problems, you should remember some of the key points. Okay, what Meselson and Stahl they did. Okay, what was the experimental tool they have used? E. coli bacteria. And actually, they were doing this experiment to find out which model of replication is correct. In the previous video, we discussed about the three models. One is conservative replication, and the second one is semi conservative replication, and the third one is dispersive replication model. And of course, now to differentiate the parent DNA strands from daughter DNA strands, they have used isotopic nitrogen. Okay, and with the help of cesium chloride density gradient, okay, they found, okay, what DNA is there. Okay, that we discussed already, and now I want to give you a perfect idea about this experiment. Okay, first they grew the E. coli in N15 containing medium. So all those bacteria they have synthesized N15 DNA because only that is available. Of course, don't forget many other nitrogenous compounds are also labeled with N15. But our focus is not on other compounds, our focus is on DNA. Okay, so that's why this is what N15 DNA. And now they transferred these E. coli bacteria into the medium containing N14, okay, isotope. So now you see the bacteria started dividing or multiplying. Of course, you know that the common method of, okay, reproduction in bacteria is binary fission, and the time required for every binary fission is 20 minutes, and in that the replication of course takes 18 minutes. Okay, so now let us say after 20 minutes the bacteria multiplied and they have isolated the DNA, and with the help of that cesium chloride density gradient they found what DNA in them, that is hybrid DNA. So in the first generation only they have, okay, ruled out that, okay, conservative model. If it is conservative model, so one normal DNA should come and one hybrid or one heavy DNA should come, or we can say heavy DNA and a light DNA should come. But here, what was the result? In the first generation, all are hybrids, hybrid DNA. That is what each DNA is with one heavy strand and one light strand. Heavy strand and light strand, and here I have used two colors for better differentiation. So just remember that red color is for N15 and blue color is for N14. Now after 40 minutes, okay, so the 2 will become 4, and one more 20 minutes, then it becomes what, 60 minutes. One more 20 minutes, it becomes what, 80 minutes. I hope you are understanding. I wrote like this: after 20 minutes, after 40 minutes, after 60 minutes, after 80 minutes. So after 20 minutes, you will get first generation. After 40 minutes, you will get the second generation. And after the 60 minutes, you will get third generation. And after 80 minutes, you will get fourth generation. And you can see now what has happened in the second generation. Four are formed, out of which two of them are hybrid and two are light DNA. From this, they have concluded that it is semi-conservative, it is not dispersive. If it is dispersive, okay, so all of them should have this N15. Okay, and the N15 and N14 should be in equal proportion if it is dispersive model. But here this N15 strands are okay, remaining same, but light DNA is increasing, or N14 strands are increasing. So after second generation, they have confirmed that it is semi-conservative, it is not dispersive. Okay, now if you go for one more generation, okay, third generation, you can see that 4 will become 8, and out of these 8, 6 of them are light DNA and 2 are hybrid DNA. And when they went for one more generation, that is after 80 minutes, they got, you see here, okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 are light DNA and 2 are hybrid DNA. Okay, like that. And I hope you might have observed one important point here. From first generation onwards, how many hybrids are there? In first generation, of course, 2 of them are hybrids. And in second generation, 2 hybrids. And in third generation also, 2 hybrids. And in the fourth generation also, 2 hybrids. That point you just remember. And so that solving the problem becomes easy. Okay. Now, for your benefit, I have made one table. Okay. You just see here. I wrote here in this column the time. Okay. 20 minutes. And after 20 minutes, you will get first generation. And what was the result? All hybrid DNAs were formed. And what is the ratio? Because all are hybrid, so no ratio here. And you just remember, they will ask you even about the strands. How many, okay, N14 strands, and how many are N15 strands. That's why I wrote here heavy strands, number of heavy strands, and here number of light strands. Number of heavy strands and number, okay, right. So uh, number of heavy strands 2, And number of light strands 2. I hope don't get confused. You see, I am talking about what? Strands. Don't forget that DNA is double stranded. So out of these four, okay, these four, how many are N15 strands? Two, very clear. And how many are N14? Two. Like that, you have to make calculation. And now, after 40 minutes, we will get the second generation. And in the second generation, what was the result? You can see there, two hybrid and two light. So what is the ratio? 1:1 ratio. And now, how many heavy strands? Two. And how many are light strands? N14 strands? Six. Okay, right. Like that, after 60 minutes, you will get what? Third generation. And in the third generation, we will have two hybrids and six light DNA. So, what is the ratio? 1:3. And how many heavy strands? Two. And how many are okay? Light strands are 14. Like that, if we go further up to fourth generation, that is after 80 minutes, two hybrids and 14 light. And what is the ratio? 1:7. 1:7. And how many are with N15 strands? Two. And how many are light strands or N14 strands? 30. Okay, if you study this table, okay, and you will be able to solve any problem very easily. So make a note of this table. So now let us see some problems. Okay. So the first one, in Meselson and Stahl's experiment, in F1 generation was found to have what DNA? Okay, light DNA or heavy DNA or both DNA. So already I gave you one table, and we know that in the first generation all DNAs are hybrid. Hybrid DNA means it is with one N15 strand and one N14 strand. So both you will find. This is the first one. And let us see the second problem. Suppose this Meselson and Stahl experiment is continued for 4 generations. Continued for 4 generations. Then what will be the ratio of heavy, hybrid, and light DNA? Heavy, hybrid, and light DNA. And don't forget that the E. coli with heavy DNA is cultured. That means actually it is transferred into the medium containing N14. So in the first generation, you got okay all hybrid DNAs, and then always there will be two hybrid DNA, and rest are all light DNAs. So I gave you already that hint. So that's why it is not 1:1:0 ratio, because you won't get okay this heavy DNA. So and the hybrid DNA is always 2, and the light DNA are 14 in the fourth generation. You can check the table. So the correct answer is this fourth one. Heavy 0, hybrid 2, that is 1, and light 14, that is 7. So ratio 0 is to 1 is to 7. And now let us look into this third one. E. coli with normal DNA, that is N14 DNA, is replicated in the medium containing N15. This is heavy isotope. Then what will be the percentage of radioactive DNA in fourth generation? Okay, now what they are asking? They are asking the percentage of radioactive DNA in fourth generation. It is a tricky question. Only thing is that don't forget N15 is heavy isotope. It is not radioactive isotope. That's why the answer is very simple. The answer is this third one, 0%. Because they didn't use the radioactive one. It is a heavy isotope, not radioactive. Okay. Now let us see this fourth problem, and it goes like this. Suppose radioactive thymines are added. Radioactive thymines are added in the medium. What would happen if a cell replicates once? If cell replicates once, or when cell divides once, how many daughter cells are formed? Two daughter cells. And what they are asking, how many of these daughter cells will have this radioactive thymine? Okay. So the options, I am reading out the options. The first option, neither of the daughter cells will be radioactive. Wrong. Because radioactive thymine is there, and the bacterium will make use of that radioactive thymine. It absorbs it. Okay. And the DNA, when it replicates, so all its nitrogen bases will have means what, all nitrogen bases means wherever thymine is there, and the thymine is radioactive thymine. So that's why this is not correct. One of the daughters, okay, in both DNAs you will find this radioactive thymine. So that's why the correct answer is this fourth one. The DNA of both daughter cells will be radioactive. Okay. Now another simple problem. If E. coli with N14 bases is allowed to grow for 60 minutes, in a medium with N15, then what will be the proportion of light and hybrid DNA? Light and hybrid DNA. Okay, 60 minutes means you see 60 minutes, how many generations? 20 minutes first generation, 40 minutes second generation, and 60 minutes third generation. And remember that here okay, the experiment is reversed. Means here the E. coli is not with N15, the E. coli is with N14. Okay, and the medium is having what, N15. So reverse to Meselson and Stahl experiment. So there actually we got, you can check that table and you will find two, okay, hybrid DNA and six light DNA were found. So here reverse. So what is that one? 6 and 2. 6 light DNA and 2 hybrid DNA. Okay, so that's what you will find here. These are some uh, problems generally asked in the competitive exams. You just make a note of these problems.